Hello my dear students, I am Prashanjit with a new video and today's topic is Drosophila and linkage. Well, we will discuss about a cross in Drosophila performed by Morgan in the year 1919. For that purpose, he chose two types of flies. The first one is having two characters: gray body colored fly with vestigial wing having the genotype B plus VZ VZ, and the another type of flies with black body color having long wing with the genotype B VZ plus respectively. Then he made a cross between them, and as a result of that he got gray body colored long wing individuals at F1 generation with the genotype B plus BZ plus. Okay, so from this cross it is clear that in F1 generation the genes B plus for color and VZ plus for wing shaped are dominant over their alleles B and VZ respectively. Okay, then he performed the back crosses. Okay, so in the first back cross, F1 generation is considered as male having the genotype B plus VZ plus, and the female is having the double recessive characters with the genotype VZ. Okay, after the cross he got F2 generation. Okay, regarding the gamete formation, it is to be noted that here the male gamete are of two types with the genotype B plus VZ and BVZ plus, and regarding the female gamete, only one kind of gamete were produced and the genotype was BVZ. Okay, so in F2 generation in the board only two types of characters were, or two types of individuals were observed instead of four, and these individuals show the parental characters only, that is gray body color with vestigial wing, and another group having black body color with long wings. No recombinant character were observed in F2 generation in the first back cross. Why? It is due to the fact that in case of male Drosophila, crossing over is lacking, and due to the lack of crossing over, it is clear that the genes present in the chromosome of the male Drosophila are linked to each other and they tend to remain together generation after generation. Okay, that is, in case of male Drosophila we may say that the linkage is strong. And what is linkage? The tendency of the genes to be remain together generation after generation, that is termed as linkage. Okay, then he performed the second cross, that is second back cross, and here the F1 generation is considered as the female with the genotype B plus BZ plus, and the male is having double recessive character. Okay, that is with the genotype BB VZ. Okay, now regarding the gamete formation, it has been observed that 4 types of gametes were produced in female. 2 of non-crossover having the genotype B plus VG, one gamete, another gamete B BG plus, and the crossover gametes, those crossover gametes will be of this type B plus VG plus, and another one is B BG. Okay, these 2 types of crossover gametes. A total of 4 types of gametes were produced by the female individual, and male produces only one type of gamete. And in the F2, in the checkerboard, it is clearly observed that 4 types of phenotypic characters were observed, and out of these 4 types of phenotypic characters, 2 types are of parental type and remaining 2 having the recombinant characters. And interestingly, it is to be noted that the total percentile of parental characters were 83%, whereas the total percentile of the recombinant characters were 17% respectively. The parental characters were more than 50% and the recombinant characters were less than 50%. It is clear that from this indication, it is clear that the genes present in the female chromosome were linked to each other, and they tried to be linked together, but the linkage was not so strong as in the male chromosomes. That is why crossover took place, and as a result of that, crossover gametes formed, and after the mating, recombinant characters were observed. Clear? So it is clear that recombinant percentile and parental percentile clearly indicates that crossing over and linkage are inversely proportional to each other. If the linkage is too strong, the frequency of crossing over is nil, and if the linkage is, that is, if the crossing of linkage is weak or linkage is not so as strong as complete linkage, then the frequency of crossing over increases, as a result of which recombinant characters were observed. So we may conclude that we may say that in Drosophila, the linkage is strong in male chromosomes and linkage is comparatively weak in female chromosome, and the genes, they tried to be linked together, but frequency of crossing over is performed. Okay, and at last we may say that the second cross indicates that 83% parentals or results of linkage and 17% recombinants or results of crossing over. It means 17% of the female gametes are result of crossover, and the distance between the 2 genes is 17 units. Okay, and we know that crossing over takes place during prophase 1 of meiosis. So it is clear to us that linkage tends to keep 2 genes together, whereas crossing over tends to separate them during inheritance. And regarding chiasmata, chiasmata are rarely formed in male Drosophila. And regarding synaptonymal complex, these complexes are formed in the females but not in the male Drosophila. So Morgan proposed that these recombinants resulted due to exchange of chromosomal parts between homologous chromosomes during meiosis. Okay, thank you.